Okay, so hi everyone. Welcome to this third OBON webinar. So we're going to start with the presentation by Raes and Philipp from the Mindre Foundation on the project endorsed under OBON called OceanOmics, Monitoring and Protecting Ocean Life. So I don't know if it's Eric, Philipp, or double act,
Yeah,
but
it will be a bit of like a mix of
yours.
the both of us. We were really pleased to be here, and on behalf of the team to present the work that we've been undertaking. We would like to start by really acknowledging everybody in the team. The whole work reflects a massive collaborative workflow from sampling at sea to laboratory analysis, computational pipelines, and as well ultimately kind of translating the science and the insights that we find with a range of different stakeholders. The ethos in our team is really kind of like linking the rate of impact of our science to kind of trustful partnerships. And this work is really a testament to that. We would also like to start by acknowledging the traditional custodians of the land that we are meeting on today here in Perth, the Whadjuk people of the Noongar Nation, and their elders, past, present, and emerging. We'd also like to extend this respect to other Aboriginal people and Torres Strait Islanders that might be listening to the presentation. So oceanomics, we're using genomic techniques to monitor marine wildlife, more specifically eDNA or environmental DNA. And we're using this as a non-invasive, rapid and scalable tool to better understand, manage and protect ocean life. Now, unlike traditional methods, which are really rooted in visual observations and where seeing is believing, eDNA shifts us a little bit more into that realm of probabilistic inference. And although we actually capture physical tiny fragments of DNA from the water or in the water from organisms, and we assign them to specific taxa and species in time and space, it still involves estimating some kind of a probability. There are some questions that remain around like how far the DNA may have traveled and how long it persists in the environment before it starts to degrade. And to tackle all of these questions, we have been partnering with the Australian government and more specifically Parks Australia. Here below you see a little movie. eDNA isn't easy to visualize, but here we're trying to capture kind of like how in holistic view eDNA can give you across that tree of life. So with Parks Australia, we've been kind of like looking into the barriers of entry of using eDNA and providing a proof of concept as well, how eDNA can be operationalized as a cost-effective tool at scale. So the tree of life here that you see spins around. We can see bacteria, octopuses, sharks, from it like 2 liters of water from Cocos Keeling Island. The focus of this partnership as well is to look into that translation to policy and help define how eDNA monitoring can be implemented into management and policy regulatory frameworks as well. So it's, as I mentioned at the start, it's a pretty long workflow from sampling at sea, laboratory, computational, and again, the translation of our findings into kind of more tangible framework. As I mentioned, to operationalize all this work with Parks Australia, we've done a number of different expeditions stretching from the tropical north here, Cocos, Christmas Island, through subtropical areas around the Abrolhos Islands, all the way down to the Southern Ocean in Hobart. We've collected samples from dynamic tropical reefs in the top 50 meters down to low energy environments in the abyssal plains here in the deep sea. Around predominantly the west coast of Australia and here in that kind of like Southern Ocean, all in collaboration with the UWA Minderoo Deep Sea team, and as well on board as the RV investigator here around the east coast of West Australia. At each sample, we get this kind of like wheel of life, this holistic insight of what we find. For 300 samples, we have this kind of, but for all the other samples, we have high depth resolution metabarcoding from fish and marine vertebrate assays. This fantastic multidimensional plot made by our colleague Marcel Eyat kind of gives you that kind of overview from these mitochondrial 16S and 12S fish vertebrates across that like long latitudinal transect where we see the southwest coast here in the southwest, Cocos Keeling Island here in the tropics, and this Abrolhos 4 years worth along the West Australian coastline, which is kind of like smidged in between warm tropical Lewin current water and Southern Ocean, and we get a bit nice kind of high tropical diversity with temperate species in there. This kind of like multidimensional plot shows you that latitudinal gradient, but also our offshore transects from pelagic species, from Fremantle to Cocos, and as well all the way down to the Southern Ocean. The message pretty much here is that eDNA allows us to track biodiversity across time and space, and it really allows us to monitor changes in the ocean and assess the resilience of our marine ecosystems pretty much at scale, and we're doing this with molecular tools. Now, people and partnerships really are at the heart of ocean sciences that Minderu has been engaged with. From Yamaji sea rangers here, we see Kira on the right-hand side, to community leaders on Cocos Island, high school students on Christmas Island over here to school that we engage with, undergraduate students on board the RV Investigator, and the many captains and crew that have been supporting our voyages. Advocacy, ocean literacy, and kind of like enabling this younger generation of ocean advocacy with new tools is pretty spectacular and rewarding as well. But we really rely on the diversity of skill sets here, from ocean advocates to scientists to policy translators to people that steer the boats. And it's a contribution from everybody that is involved. Data sovereignty, as we have seen, and ownership, especially with communities, is really important with everybody that we have engaged with. And everybody expressed as well a really strong interest to kind of like have a first look at our data, they call it, of course, on their country or in their lagoon or river, and to kind of like access it and understand what is happening there. And so to kind of solve that problem,
Thank you. Hi, I'm Philipp. I'm the computer person, so I'll be talking about the computer stuff. To solve the problem of making all this data come alive and be useful for stakeholders, we've developed a bunch of solutions I'll be talking about. The first one is the Marine Parks dashboard, which you see there, screenshot of on the top right. It's an online dashboard developed together with AP Interactive out of New Zealand that lets you explore various aspects of the ODR associated data across various assets. So for example, you can go in and explore by park, you can explore by species. There's a chatbot in there about which I will talk about in a second, and then there's an explore the unknown visualization about which I will also talk about in a second. But yeah, this is one of the first solutions we have for, again, making this data come alive and useful to the public and our stakeholders. Thank you. So, for example, this is the search by species view there on the top left. You can either search by species names or taxonomic group. We just came up with a bunch of regular groups, and or by IUCN conservation status, for which there's an example here on the right. And there's a bunch of high-profile species we've added to, so you can search for them. Thank you. And then here's, for example, an example for threatened species, then displayed on a dashboard. Where did we find the threatened species? You can here see how our species sightings are split up by IUCN status, and then up here on the right, you see kind of where are the hotspots for these IUCN species in our data set visualized on the dashboard. Another aspect of the dashboard is this check with the data function. That's the thing we developed on top of external AI models. In this case, it's Anthropic's Claude. That is a model that lets users ask regular English questions. The model then writes Python code. We run the Python code, and the results of the Python code are returned to the user. It's kind of like a way to have people get answers to their questions that are not answered by the dashboard. It's reasonably powerful. We're still developing these kinds of approaches where you have like AI and large language models work with the data that we've generated, again, to get more out of the data and commit more to make it come alive a bit. There's a lot of work we're putting in there on data curation, where we compare the eDNA data to various external databases, like in this case, ALA, Fishbase, and Acromap, so you can curate these sightings too. And there is a explore the unknown function, which I will talk about on the next slide. Let's have a look at that. Now, first of all, this one. This is a report function we've also been working on, which we're about to launch probably this week, where we automatically generated reports for every site we sampled at. So we get like one report for every bunch of replicates. In that report is a, first of all, split by IUCN status, a gallery of species, which one did we find? There is a AI-based species summary in there where we gave the species list to another external AI and just told her, please pull out the interesting species, species that are interesting to regular people, and describe a bit why they're interesting. Then there's phylogenies in there and things like life history related figures and things like that. So people, again, this makes the data come alive a bit better. And the other thing I just mentioned was this explore the unknown function. This is the thing I've developed based on DNA-BirdS, which is a DNA-based large language model. It's open source. We fine-tuned this using all the ASVs we gathered for 12S and 16S, so about 20,000 ASVs each for 12S and 16S. And then this is a visualization of what the model has learned, clustered into 2 dimensions by clustering method called TSNE. Every dot you see there is a ASV, a unique ASV, and they're colored by family, by taxonomic family we could assign to it. So some of these families cluster nicely together, like the Phothidae down there, for example, or the Extraquotidae a bit to the right. The gray dots are ASVs for which we don't have any taxonomic labels. They're currently unknowns, but we will eventually have labels for them. But this is kind of like a nice first step towards trying to map the diversity in a site without knowing everything about the taxonomy. So kind of like a first step towards taxonomy independent approaches. Here's another, this is the same as what the model has learned, same figure, just split up into sites. This is from this Cocos-Keeling transect from Fremantle to Cocos-Keeling, and you can see that obviously every site is a bit different, but we keep on finding these unknowns. And currently working on building essentially biodiversity gradients based on these plots, so based on what the model has learned for every site, and then comparing sites directly without knowing too much about the taxonomy in these sites. This is the last slide. Miwa is here today, so I won't be talking too much about FAIRY as a standard, but all of our data is now aligned with the FAIRY standard. We have started to upload all the data we have generated to various open repositories. Like we have a few runs on the eDNA for which I have to go back and redo everything. We about next month, we're going to start to submit all of our biodiversity sightings to OBON. Our code for the pipeline is on GitHub. What I just showed with the embeddings with the DNA-BirdS for eDNA is also on GitHub down there. And then there's various AI-based tools also on the GitHub, which I haven't talked about today, but which are also interesting to look at, like, for example, this Curate with Claude. And with that, I will return to Eric.
Wow, amazing. Thank you, Philip. The Australian marine estate is pretty large. If we go here from the top of Western Australia all the way down to the south, that is the equivalent of going from the top of Norway pretty much to Portugal. So scaling the collection of eDNA is not a straightforward kind of task. And for that, we've partnered with CSIRO, the National Australian Marine Science Agency, and we're deploying an eDNA sampler on its national research vessel, the RV Investigator. For the next 3 years, it will be taking a sample every hour while the ship is conducting expeditions around Australia. All the samples will be coming to the Minderoo OceanOmics laboratory, where Shannon Corrigan and her team will be sequencing 4 metabarcodes and potentially also some shotgun data from the samples that are coming from the Investigator. That is kind of phase 1 up to 2027. Phase 2 will then see this eDNA auto sampler be implemented in ships of opportunity. And this can be little tugboats, tourist boats, or you name it. We will be implementing this kind of automated samplers on board these ships of opportunity. And that's really an example how we can scale and integrate, kind of interweave eDNA as well with multimodal data, especially on the Investigator, which will be collecting a whole suite of different physical and chemical metadata. And that kind of like linking all different data sets together will really allow us to better monitor and better understand our marine ecosystems. Now, while drones and automated samplers allow us to scale, some music is playing here in the background, which you can hear it. Sorry, I think we have here some music playing.
Ignore that.
I will ignore it. While drones and automated samples allow scale, people remain at the heart of every decision, at every action, and every discovery in ocean stewardship. Minderoo is also really proud to be able to support the second phase of the eDNA UNESCO expeditions in all the marine world heritage sites. And this is really coming back to the idea that we now will be mobilizing, engaging communities, local rangers, and especially young citizens, giving them tools to do hands-on science and conservation with cutting-edge yet accessible technology. Another kind of project that we're really proud to support is the Citizens of the Sea program, where we are funding the equipment and the analysis of torp eDNAs, which are torpedo kind of eDNAs, which sailboats drag behind their yachts, filter eDNA in like real time, and then it will also be all kind of like sequenced. And again, this project really enables citizen science to collect data at scale at a fraction of the cost from research vessels. And here I think we had like it is loading now, a movie kind of like showing all these UNESCO sites. And then it will also be showing our sites that we have been collecting under the Parks Australia partnerships, the Ocean Discovery and Restoration Program. And we're also really proud that we'll be able to support and fund part of the BioGoShip initiative. And this BioGoShip initiative really uses state-of-the-art research vessels and really linking in here, same as on the investigator trip, physical, chemical parameters that will allow us kind of to build up that ecosystem lens. And then we will be starting to interject eDNA into that as well. Ultimately, again, to give us a better picture of what is happening in our oceans. And with that, it's a long list of acknowledgements, definitely starting with Dr. Andrew Forrest and Nicola Forrest for funding this whole endeavor. Many people at the University of Western Australia and other institutes in a long list of people and institutes and logo. Thank you very much.
Thank you very much.
Thank you, Eric and Philip. That was great. Yeah, sorry, I was just monitoring the waiting room. Yeah, really great overview. And you actually mentioned and showcased quite a few other initiatives that are either already OBON endorsed projects or hopefully in the process of becoming endorsed. So really nice way of showing the connections across OBON as well. I have a question about the with the Glombo project and the plan of doing putting the auto sampler on ships of opportunity. How much sort of work will be involved for the crew or whoever's going to be deploying the auto sampler and collecting filters and things?
Yeah, so we're making this as straightforward and easy as possible. Pascal Crowe, who is the lead engineer at CSIRO, asked the engineering team on the investigator, how much time do you indeed have to help out? And the engineering team said 5 minutes with a cup of coffee in our hands. And that's kind of like where they're like working towards to make it really straightforward with filter cartridges, the same ones that are being used at the UNESCO eDNA expeditions program. Which are encapsulated. There will be a preservative injected, which is room temperature stable. And the idea is that they will come in 24 cartridges. Like the cartridges will sit in like a slot and they will like slowly move around. So every sample will be kind of preserved, and whoever's on the ship can take out the cartridge, 24 in one, and slot in a new one. They're also working to have this kind of like 24 case of cartridges stacked. So you could have like 2 or like 3. And then it would be a swap over of like every day or every 2-3 days, really minimal effort.
Okay, great. And how is the auto sampler deployed? Is it sort of attached to the side of the ship or is it dangling?
Yeah, so there's a bit of like, so that's why we're starting on the investigator and probably branching out to some other research ships that have dedicated inline flow through lines that are coming at probably at like the hull, which kind of like try to be for scientific instrumentation. That will be like the goal to start with. They are already looking at a little ferry in Hobart, which goes every day around with like tourists, and implementing that into the engine room, and a glass bottom boat up in the Great Barrier Reef that will go around a coral reef. And there it needs to be kind of like tinkered a little bit more. The glass bottom boat might be with like a little sump pump on the little ferry in Hobart that might be taking water from the raw seawater that is cooling the engine with kind of like a pre-filter screen. So there are some various options, starting kind of like research vessels dedicated inline, cooling water for the engine, and then like an additional kind of like hose, kind of with a little.
Okay, great. Thank you. Are there any other questions?
Yeah, hello, Siddesh here. So I just had a question regarding the profiling. So do you go for vertical profiling also with the same, along with the like horizontal profiling of the water sample?
Most of the time, we've been collecting sea surface water in the top 50 meters, either with like our standard well-trusted Niskin bottle or a kind of more high throughput filter sampler from Ocean Diagnostics that can filter at depth down to 400 meters. Mostly we have been deploying it to 50 meters. When we are going on the dedicated research vessels, we have the rosette, as we can see behind Sophie on the ship, on her background, and then we can take water at any depth pretty much, and we do like depth layered. We have a number of expeditions. We have samples from the surface, 100,000 up to 5 and to 6,000 meters. The samples that we collected in collaboration with the deep sea laboratory, they have been collected with dedicated deep sea landers that are deployed. We have Niskin bottles attached to that, which are open, and there is a galvanized link that will erode after 5 hours, and it will shut the Niskin bottle close, and the lander will drop its weight and will slowly come back to the surface. So that's kind of our like depth resolution.
So are there any specific number of samples that you collect from each location in a particular depth?
The surface samples were always 5 replicates at one site, and then kind of it depends on the length of the voyage. The expedition that we did on the East Australian current, that was a dedicated 3-week, a little bit longer voyage, and we collected 1,800 samples on that one alone. So it's we've done trips to the Abrolhos Islands on the west coast. We normally go there only for 6 days, and then it's like around 200, 300 samples that we collect. So it like varies.
Yeah. So that's what. So that's it. Yeah. Thank you.
Okay, thank you very much. So you kind of nicely introduced the transition to the next presentation by mentioning Miwa's project, which is making eDNA fair. So Miwa is from CSIRO. And when you're ready, if you could share your screen, that would be great.
Oh, I'm going to try that. Hang on a sec. Sorry.
Okay, I can see your.
Yeah.
There we go.
Oh,
Yeah,
is it?
that's
Good.
great.
Okay.
Thank you.
Cool. Thank you. Thank you very much for having me today. My name is Miwa. I work at CSIRO, and today I would like to present about the project that I've been doing for a bit over 2 years to make eDNA data fair. And I'm glad Eric and Philipp went before me, and then they introduced about their amazing projects and upscaling the samplings and then collecting all sort of samples and then producing lots of data. And then they also touch a little bit about this fair eDNA data format. So I'm going to go a little bit deeper in that. Fair principles. So the FAIR stands for findable, accessible, interoperable, and reusable. I might try to use a highlighter. Sorry. Can you see my pointer? So here is that's the acronym of the FAIR. And then we also have an acronym for FAIR eDNA, which we call now FAIRY. So with this project, we are basically trying to make the thousands and billions of eDNA data out there now in a standardized format so that we can collect them together and make a huge eDNA biodiversity data set, and then we can reuse it for future analysis. And to do that, we developed a FAIR eDNA guidelines with a number of eDNA researchers and database managers, journal editors were also involved. So it's a very big collaborative initiative with number of collaborators, and those collaborators you can see in this authors list here. So these were the people, these are the people who helped and develop these guidelines together. But there are also a number of others that were involved in the development, and also more newer coming in. So I would also like to acknowledge them. But basically, so this paper was published mid this year, and then it contains quite a lot of information, which I'm going to go through very quickly. It also has a website here that I would like to highlight. And those websites basically contains all the information from a journal paper, but also it has more updated stuff, because we are continuing to improve and then develop and update. So all these updates would go onto this website. So this is the place that I would like to direct everyone to watch in that space. Okay. So the guidelines have several contents, and the first one is the data components and the formats. So with the working group, what we worked on at the beginning is to define what data components are important to share, and then in what format. And then, sorry, my slide is not working well. It's moving by itself, but we can move on anyway. Then the second part is the metadata checklist. And then it's a very comprehensive metadata checklist. It basically has a list of terms that should go into as a column headers in the number of metadata or data components that I introduced earlier. So it is a comprehensive one, because we want to make sure that the metadata is rich enough and it has all the information required for data reusers to understand how the samples were processed, and how data were produced and then screened and all sort of things. And those information are important because standardization of eDNA methods is very challenging because of number of reasons. Sometimes the fundings are limited and the environmental conditions are different, and the scope of the study is different as well. So the data reusers have a different scopes for their own research. So the understanding how the samples are processed and data were screened, those are really important. And when we're talking about thousands of eDNA data, we cannot screen those things manually. So having that in a machine readable way is also very important. And that's why we produce this checklist with a controlled vocabulary, and then we're trying to guide eDNA practitioners to use this standard. And this checklist terms come from existing data standards. So there are very strong established data standards. For example, there's a biodiversity data standard here called Darwin Core, and MIXES is a molecular data standard. GBIF, for Global Biodiversity Information Facility, also developed the DNA-derived biodiversity data extension. So they are all very relevant for eDNA data. But there were some gaps that we identified to fully explain eDNA workflow, and that's where we created a new terms, and that's why I added here, plus more. Then once we developed those first two components, the next thing we did was to put a step-by-step guide. And this is what you can see here. So from 1 to 6, it starts with a template generation, and then all the way to publishing data, and then linking the published data links such as DOI into a scientific paper's data statement. And where to publish data is described on here. And basically we're encouraging eDNA practitioners to publish their data in established databases such as GBIF and OBIS if it's a marine data, and INSDC for sequence data. And the reason is because those databases contain billions and billions of other type of data biodiversity data or sequence data. And if eDNA practitioners put our data in there, and that's where our data can really become fair along with other large data set. And the last contents here is the tools and the scripts. So along this fair eDNA theory practice step, we developed few tools to help eDNA practitioners to make the process easier, quicker, and also more accurate. So for example, the first one here is a Fairy-ator or Fair eDNA template generator. And it's an R function to generate templates based on the study design. The next one is a Fairy-Fire. It's a metadata verifier. And the last one here is a Fairy to MDT. MDT is a meta-barcoding data toolkit developed by GBIF. And the Fairy formats and MDT format are slightly different for reasons. We've been working together from the beginning, but we just for a few reasons, we have a slight difference. So this tool would reformat the data for that to make it ready for MDT submission. And then Fairy guidelines. So now that this guideline has been published, we've been running multiple workshops for to encourage the community uptakes. And we ran several workshops this year. Excuse me. And then number of eDNA labs and researchers have adopted Fairy guidelines and formats and checklists and things like that. So I'm really excited about it. The Foundation is here, as Eric and Philipp explained and presented just before me. There are thousands of amazing eDNA data are now in a Fairy format, so I'm really excited about it. And NOAA is another collaborators from the beginning of the project, and then also they are formatting their data in the same way. And IMOS is an integrated marine. I'm sorry about this slide issue. IMOS is an integrated marine observing system, and I just recently joined IMOS, and then we are now developing an eDNA sub-facility. It has been actually, the sub-facility has been launched recently. So we are going to produce a lot of eDNA data from all around Australia throughout the time series, the extended time series. And yeah, all the data is going to be in a Fairy format and published to databases. So there's a lot of community adoptions now. And then the other component here on the right-hand side is a scientific journal. So we've been working closely with the journal editors because we believe that journals have really significant impacts on the practice of the scientists. So for example, open data practice is now a very normal thing. Everyone makes the data open because they mandated it a while ago. But then it's open, the data can be open, but not entirely fair. So they do recognize the gaps, and then they realize that, we all recognize that domain-specific guidelines, fair guidelines is important because to make it really fair, make it all interoperable and reusable, every domain has a different protocols, different type of data. So this kind of domain-specific guideline is really important to implement. So that just happened, as you can see here in the highlights, with the eDNA journal author guidelines. They included these Fairy guidelines as a recommendation. And then we've been also working with Pensoft. They have an MBMG, the Metabarcoding and Metagenomics journal, which is also very relevant for eDNA field. Okay, so the next step, what we've been working as after we published the guideline, is to develop the eDNA metadata checklist within GSC and TADWIC frameworks. So GSC is a genomic standard consortium, and TADWIC is a biodiversity information standard. And both of them are very relevant for eDNA data, and they are established metadata standard organizations. We've been working with them from the beginning, and we've been implementing their terms if that already exists. But as I mentioned earlier, we also created a lot of new terms in our checklist. So we decided to work with them, and the reason why it's very important to integrate within their standards is because these databases on the right-hand side, they use the Darwin core from Tadwig and the Mixes from GSC. They use these terms. So this collaboration and integration will bring a direct bridge, direct link between the ferry and these databases. So the first thing we did is to formally establish the working group under these two organizations. And you can see the members here are actually identical because we are working together and developing both standards concurrently, because there will be a lot of overlaps between those two checklists. And once these checklists are established, then what we're going to do is to map these different standards using SCOS or SSSOM, this kind of mapping standards. Because even though there will be huge overlaps between these different standards, they won't be identical because the scopes of their standards are slightly different. So we're going to use this kind of mapping, and then we're going to develop this kind of tools to make sure these mappings and then all this reformatting can be done easily. And I mentioned this ferry to MDT is already up and running. And then a ferry to NCBI was developed by NOAA recently. And then along the way, similarly, a ferry to eDNA, this European Nucleotide Association. That's under development. And I would like to highlight that this kind of development of scripts are happening outside of the ferry working group, which is really exciting to me because the community uptakes the ferry format, and then they realize, okay, there are tools that we can use, but then there are some gaps and we need those tools for our workflow. And that's how NOAA just developed this ferry to NCBI, and they didn't even tell me about it. And after they developed, they told me. And then I was talking about it with Philipp from Mindaru, and then he said like, oh, we actually can make this ferry to eDNA because he already has a very similar script to submit to eDNA, and then they are adopting a ferry format. So this kind of things is happening just spontaneously in the communities. And then I would like to make this pragmatic submission part all implemented and then make everything automated. So here, I mentioned these ferry practice steps, and some tools are already available. And then another tools that I am working on, sorry, I mentioned about this NOAA team. So they made a ferry to NCBI. Also, they have these two other scripts that the ferry formats are all in there, and then they are using it in their workflows. Sorry, I just got the order wrong. I mentioned about this ferry to eDNA, the conversation I had with Philipp in Mindaru. And the last part here is the BBOP. It's a better biomolecular ocean practice, and it's another project within the OBON community, and that's just through OBON webinar last year, just like this one. I found about this BBOP project, and then we decided to work together because there's a lot of overlaps between our projects, and now we're talking about developing some tools and scripts to format the ferry table metadata and then produce a BBOP standard method description, or the vice versa as well. If someone follows the BBOP protocols description, then from there we can extract the terms and put that into table format, the ferry table format. So something like that is also along the radar. And now recently we created within the ferry initiative, we created a new working group specifically for coding, and we're trying to parse all these bits and pieces of scripts under R and Python packages, because at this moment it's a bit dispersed. Some of them are R functions, some are Python package and Python scripts. So we're trying to unify them together to enhance the automation. So that's where we are at. That's all from me now. Thank you very much for listening to my talk. And I would like to acknowledge all the people who've been helping for this project a lot, especially those FAIRY working group and that week GSC eDNA task group that we've been working a lot this year, and then this newly developed FAIRY coding group as well. Thank you.
Thanks very much, Miwa. It's very exciting to see all these developments since you presented around this time last year, when I think you'd been recently endorsed by OBON. Are there any questions? No? Well, it's really great to see all the collaborations and how you're working to make things easier for eDNA scientists to make their data fair and not have to submit it to multiple different databases. So that's very exciting, and looking forward to having more discussions on this in Hobart next week.
Yeah, definitely.
Okay, so thanks again. So I think Ioulia is here, who will be
Yes, hello.
presenting on OBON, sorry, not OBON, EMOBON, from the European Marine Biological Resource Centre, EMBRC.
Okay, I see the sharing is starting. Thanks for sharing. Okay, great. Thanks for sharing the slides. That helps a lot with my internet connection. Hello, everyone. Good morning and good afternoon. My name is Ioulia Santi, and I represent the European Marine Biological Resource Centre, which is a European research infrastructure. And here I'm presenting our genomic observatory EMOBON, which stands for the European Marine Omics Biodiversity Observation Network. So in the next slide, we have a few introductory points just to remind everyone and establish the context a little bit, and also to explain where our initiative came from. Marine biodiversity is one of the core components of the ocean system. So fundamental that many times it's what it sustains an ecosystem. It's tightly connected to human society, and we sometimes forget that essential services like oxygen or food or the connection to the economy are tightly linked to the marine organisms themselves. And of course, the integrated ocean management vision that's in the blue economy that we hear a lot about it is actually balancing the economic development and the protection of the oceans. And it requires information on biodiversity and its integration into those management activities. And recently, this understanding is going beyond the scientific community, and the protection and restoration of biodiversity is emphasized by several different policies, at least in the European level that we are working at. However, the capacity for biological observation is not as standardized or elaborate as it is for the other ocean scientific disciplines, where it's fully operational. So here we all know that sampling can be demanding, and it's also challenging and expensive. And the dynamic of the biodiversity is making everything difficult, how we capture the representative snapshot and then establish all those consistent baselines among the organisms. And then, of course, this connects to that we don't have standard operating procedures for data generation. And to other challenges like long-termity of the funding schemes, and also on the data that we don't have standardized methods for data collection for verifying our data. So yes, it's urgent that we organize marine biodiversity observation, that we connect with each other and go for coordinated efforts. And in the next slide, our organization, based on those points, responded to this challenge for coordinated genomic observation. it does fit to our strategies of providing access to ecosystem biodiversity, following and implementing inclusive and global standards, also exploring marine biodiversity and enhancing the global observation. So it fits really well with our objectives and our missions. I should also tell you that the EMBRC is, well, it's a research infrastructure, but perhaps this doesn't mean anything outside of the European context. But I need to say that it's composed of different institutions, marine institutions, that are distributed in many different countries in Europe. And then there's the central coordination from the headquarters in Paris. And in the next slide, please. Yes, so we established OBON, that's the European Marinomics Biodiversity Observation Network, aiming to employ the genomic techniques to access biodiversity at the European coastal seas. So at the moment, the network is based on 21 institutions, partners of the EMBRC in 10 different European countries, and it includes 17 different sampling sites for the biodiversity observation. In the next slide, so we describe all procedures and strategies, and I'm going to go into details in a moment. But before that, I would like to stress some of the characteristics that make OBON impactful and important at the European context. So starting with that it is a long-term initiative and that we are now at the 50th year of producing continuous data, and that this scheme is including the temporal component of the dynamic biodiversity, and it supports the continuous data generation that goes beyond short-term funding schemes. Then OBON is also producing comparable data using standardized methods from collection to publication, and this increases the potential and significance not only for original first-hand research, but also to support large-scale meta-analysis. Then the framework builds, it expands actually beyond collecting samples and analyzing the data, and we work a lot on how all the procedures and regulations are recorded and open and shared with everyone. And of course, we provide full data provenance for all the data that are produced. Of course, all these procedures are working projects and they are evolving along with the project. And yeah, last point is that as this event also demonstrates, OBON is part of a global system, OBON, and it's eager to synergize and work with other projects and to support the openness of biodiversity observation. So in the next slide, we can see some details for our operations. We began sampling in 21, and it takes place regularly, but differently for the different habitats. So for example, for the seawater microbial community, we sample every 2 months. In addition to the seawater, we are collecting samples from soft sediments, targeting microorganisms, myobenthos, macrobenthos, and also hard substrates where we target metazoa communities. And of course, we collect reads metadata to perform the centralized data management. And we are incorporating the MIXIS checklist and the Darwin Core standards. Also our analyses are centralized, so all samples are processed at the same way in one centralized facility. And we also follow a coordinated approach to facilitate the sampling permits and the access and benefit sharing that is following all the biodiversity samples. In addition to that, we also collect biobanking samples for biobanking. And at the moment, we have more than 1,200 samples that are biobanked. And of course, all our protocols and data procedures are open and anyone can find them. So either in our website or in the OBPS, I can give all those resources at the end of this talk. So then in the next slide, that's just a diagram showing that the 3 different modules are sampled at the different stations. This is at the 17 sampling sites. And then all samples are collectively sent to the Genoscope National Center for DNA extraction and sequencing. So all the methods are standardized and data are fully comparable. And then we also have at least 2 replicates, technical replicates, that are stored at the stations. And this is for later exploitation on projects or collaborations. Then in the next slide, we can see the flow of the data, which is that obviously our raw data are going in eDNA, and then our process data in Magnify. This is a work in progress because we have the same, so we work together with Magnify to make it possible that data that are analyzed outside of Magnify, when they meet the standards, they can also go on the Magnify database. And then the metadata go in Eurobis and GBIF, and from there to Immonet Biology and Editor to make it to the digital twin of the ocean. And this is also work in progress. We have data publications to announce the datasets that are published. And then, yes, we have all those data products and analytical tools that are developed in the different projects and that are connected with Eurobis and Immonet. And with all that, we want to focus on data reusability, research, innovation, and policy, not just in our community, but of course, everywhere in the global level. Then in the next slide, yes, so this was, let's say, what OBON is. So now I can highlight some of the impacts and achievements that we have in this last year. So in the next slide, please. Yes, we had our first data release last year. No, actually, that's the numbers. Yes, that's OBON in numbers. So, and some of these are very impressive, and a lot of people like to see. Also, our funders ask for them. So, since its creation, we have collected almost 5,000 DNA-based samples using those standardized methods. And you can see also that about 2/3 of those are sequenced, and we have also the ones that are stored. So 3 out of 5 are stored for the long term to be assessed later in different projects and different initiatives. And then in the next one is some of the recent works that we are doing. So in the next slide, please, we can see the data. Yes, that's the first data release that we had in 24. So those are in eDNA, and there is also a data publication that is describing everything. And there's also a different one for the, so the first one, the one I just mentioned, is for water and sediment microorganisms. And then there is another one that is describing the hard substrate communities, that's metazoa. And again, they are in eDNA, and there is a data publication to describe them. So we also have now the second data set on eDNA, and we are preparing the data publication, so it will probably be out by the end of the year. Yes, and another thing we did in 25 is that we established science working groups to actually start tackling those data and have a coordinated effort on analyzing the data. Yes, and then please go to the next slide. So here we have colleagues working on the hard substrate metazo communities. They are working on non-indigenous species, and this is a publication on which they detected and found 63 non-indigenous taxa at coastal locations in Europe. And they emphasized that their presence, the species presence, was regardless to the high maritime traffic that was noted in some collections. Then there are some concentrated efforts using the data in our community. So one ongoing work is focusing on benchmarking metazoa inferences from the Zootken metagenomic samples. So they are actually trying to fish out fish genes from the metagenomes and then comparing it to metabarcodes that they are collecting locally. And another noteworthy work is focusing on using the metagenomics data to map the metabolic potential of the eukaryotic marine plankton. Also, this month, actually, a week ago, we participated in the 9th Ocean Hackathon, which took place and was organized by the University of Évora in Portugal and the Marine and Environmental Sciences Center. And there was a team in our network that worked on an interactive map to track the genomic biodiversity. So the work is actually boosted and initiating in the hackathon, and now we are expecting that it will have a life of its own and continue. And the next slide, please. We can see, yeah, some of the challenges that we've been facing in 25. So one of the active and ongoing work is that our data, our analyzed data, and the actual biodiversity information are going in EuroBIS and then OBIS, along with all full provenance information that we are collecting in the metadata. So this is done in the framework of the DTO Bioflow project, which as an ultimate goal has that the biodiversity ends up in the digital twin of the ocean. And so this has proven to be a demanding task. We had to improve QC controls, create scripts from scratch, and we are working a lot with the OBIS team. And when this work is done, it will pave the way for other metagenomic data to be easily incorporated into OBIS, sorry. Yeah, so this is work in progress. It's demanding. However, we expect to have some cerebral project progress in the next year. Then another challenge we are facing, but it's not included here, it's that it connects with everything that we are doing, and it's that there's a bottleneck that is created during the bioinformatics analysis of the data. So for shotgun metagenomes, it takes us 2 to 3 days to fully run and analyze one sample in a supercomputer. So therefore, we are lagging behind on the matter of the analysis of the samples, and then all tasks following this are connected to this bottleneck. Yes, so yeah, and the last challenge, let's say, is that yes, we are facing a reduction on the budget, and that's very often what we are hearing lately. It starts from this year, and we are looking to limit our operation, but still be relevant to the scientific audience and not reduce the quality of the data and operations. So yeah, the last slide is because I wouldn't like to close with the challenges, but with some considerations and thoughts. So yeah, to successfully obtain this long-term funding, it's necessary to have clear interest from stakeholders and then also connection to applications. So for genomics, some applications that we have identified and are targeting us is, okay, first, the easy one to support science. So provide work on detailed genomic series for marine microbiome, and then that's in an expanded geographic area. And it can be implemented also further with applying the essential ocean and biodiversity variables framework by COOS. To support policy, it connects, for example, with the 30 by 30, which refers to the global target to protect at least 30% of the world's land and ocean by 2030, the Green Deal, the Ocean Pact, and there are specific mentions in those policies for biodiversity observation. Then to support blue economy, our data directly support aquaculture, ocean health, and biodiversity components for a holistic and sustainable management of the system. However, it's not yet obvious how those data directly connect to aquaculture and ocean health, and this still needs to be made obvious by our community. One thing that is obvious, at least, is that biodiversity is becoming an effective part of the digital twin of the ocean. And that's something that the communication is working well. So these perspectives are shared by the scientific community, but we still miss other stakeholders like policy, other scientific communities, enterprises, and the general public to piece together this puzzle and to move to a next enhanced level of operation. Well, that was all for me. The next slide, I just want to mention that this is an initiative by many colleagues behind this. In EMBRC, we are coordinating the network, but there are many nodes and many people around Europe that are contributing to make OBON what it is. Thank you very much for your time, and I'm happy to be part of this seminar.
Thanks very much, Ioulia. That was a great presentation. I have a question, but I want to give the opportunity for anyone else to ask a question. It was more of a comment, actually. Just really interesting to hear about the work you're doing with OBIS on getting metagenomic data into OBIS, and hopefully we can hear a bit more about that from, I think Christina is attending the OBON meeting, and we'll have a
Yes, and
session
she's also
on
our
data.
yeah, and she's also our data scientist, so it's mostly her
Okay.
work that is doing that. So she's the appropriate person to discuss this further.
Okay, that's great. Yeah, because we'll have a session on data and OBIS and getting data into OBIS. And also sort of linking with or talking about getting data AI ready. So the perhaps the work that you're doing also with digital twins and Edito is something that she can share as well in that context. Is that quite preliminary, the work on getting eDNA data sort of ready for digital twins? Or
No,
do
it's not.
you...
It's not preliminary. It has progressed a lot. I think in 26 we'll actually have the first data set in OBIS, passing all the QCs and have all the script ready to share. And perhaps after that, I think we'll need some enhancing the scripts to make it available to everyone. So it's not preliminary. We have progress and we have details to share about that. Then once everything is in OBIS, it automatically goes into Emonet and it's part of the digital twin.
Okay, that's great. Well, thanks very much again. We'll move on to our last
Can I
presentation.
ask one question if there's time?
Yes, sure.
Yeah, okay, thank you. Yeah, very, very nice talk, and yeah, I'm fascinated with the project. So you mentioned that, sorry, along my project on the FAIR eDNA side, you mentioned a lot about data sharing, and then you share all the protocols, and you use mixes and Darwin Core. So I was really interested to hear more about it. So I wonder, did you find those mixes and Darwin Core terms were like, could you find all the terms that you needed to for your data set from those data standards? Or if not, I would like to see which terms were not, because that's what we are working on to fill the gaps. And then, yeah, I would like to see what your data is like.
Yeah, so we started with mixes and then checked what is the same in Darwin Core. So in our case, I think almost everything is covered with the mixes list in Darwin Core. We have some special terms that reflect our operation. So for example, because we are shipping samples, we want to know the date of the shipment and the date of the arrival of the samples. So these terms we have to create, but those are relevant just for us. And then also we had to create many different terms for temperature, because we have temperature at the observatories, temperature at during shipment, temperature at the sequencing center. So this was a bit tricky because we had to, we had the same, let's say, value and the same thing that is temperature, but we had to separate the terms to reflect exactly what its temperature is. Yeah, but other than that, I think everything is covered, especially in the molecular path, so the laboratory part. I think that's covered pretty well. Yeah, and also from your presentation, I was thinking we should connect and check what the eDNA, the FAIRE guidelines suggest, to see if we are there and if there's anything to gain from your work.
Amazing. Okay, thank you. Yeah, so those kind of little details, like everyone has a different protocols, and in your case, you ship the samples, and then we didn't really cover that part. So that kind of any feedback would be amazing to hear. And yeah, we should connect after this talk.
Yes, yes.
Yeah. Are you going to Howard next week?
No, but a colleague is
OBON?
there. They are representing me. I can give her your name and details, and you can connect with her. So we are on the same page.
Amazing. Okay, thank you.
Yes, thanks, Miwa. I was also wondering about that, if Emobon was sort of aware of the FAIRY data standard. And yes, there'll definitely be opportunities to connect with Christina next week. So looking forward to that. Okay, so our final presentation will be from Lucia Campese, talking about the NEREA Observatory in Italy.
Yeah.
If you'd like to share your screen.
Yes. Okay,
Okay,
you
that's
see it?
great. Yeah,
Okay.
perfect. Thank you.
So good morning, well, good afternoon, good evening, everyone. I'm Lucia Campese from the Stazione Zoologica Anton Dohrn in Naples. And I'm very happy as well to be able to present today a little bit what we are doing with the NEREA project, which started in 2019 and is part of OBON since 2022. NEREA was born to merge originally two main ideas, two main projects. One is the long-term ecological research Mare Chiara, which is one of the longest plankton time series since 1984. And on the other side, the experience with Tara Ocean's data and Tara Ocean's protocols in terms of advancing the technologies and integrating the omics into the traditional methods for monitoring that we had in the long-term ecological research Mare Chiara. So NEREA is the Naples Ecological Research for Augmented Observatory. And this is a project that has three main objectives. So on one side, we want to advance and augment the long-term research on marine ecosystem dynamics, given the knowledge that we acquired in all these years of traditional monitoring, but also implementing it with new technologies. But we don't only want to perform monitoring, but we also want to investigate specific processes through targeted field studies. And we also want to link field observation with lab experiment and advance the crosstalk between the two disciplines, let's say, experimental and observations part. So the study site is the Gulf of Naples. Other than the long-term ecological research Mare Chiara site, which is here 2 miles off the coast, we also added two other sampling sites. One is the NRS here, the river mouth of the Sarno River, which is one of the most polluted rivers in Europe, where we sample seasonally. And one is the NRC, which is a canyon near the island of Capri, which is a deep canyon where we go sampling twice a year, while in the Mare Chiara station we sample each month. We also sample different depths, especially at the Mare Chiara station we sample the surface and at 40 meters. And we sample at different depths in surface and very deep samples also in the canyon Dohrn. So the strategy and the workflow involves the size fractionation sampling with different filters, and we have nets and the rosette equipped with the CTD, and then in the lab we do chemical analysis, flow cytometry, sequencing, and so on. So we employ nets and Niskin bottles according to which is the size fraction of sample of plankton that we want to collect. So we have phytoplankton nets and zooplankton nets, CTD equipped with the rosette. And at sea, what we do on board is this the same protocols that we took from the Tara Ocean's experience, where we filter samples of water on different filters. So we have a 20 micron of nylon filter to collect the 20 to 100 size fraction. Then we have a 3 micron filter to collect the 320, so the nanoplankton. And then we have the 0.2 microns filter to collect the 0.23 size fraction, so the picoplankton, which is enriched in free bacteria. And we also collect what is the outlet of this process by precipitating the viruses. So we have the whole span of organisms with this approach. So this is how it looks on board. So the tank, the small filter here, the peristaltic pump, and the 2 tripods with the other 2 filters. And then we collect the filters and we immediately freeze them in liquid nitrogen. And once in the lab, there will be the extraction of molecules from those filters, either DNA, RNA, proteins, and metabolites. And then we go ahead with the DNA and RNA sequencing and with the liquid chromatography and mass spectrometry for proteins and metabolites. So this is done together with all the physical and chemical parameters, but me personally, I'm mainly involved in the metagenomics and metatranscriptomics. So this is what I'm gonna talk more about. So we obtain at the end all the plethora of omics data. So amplicon sequencing, metagenomics, metatranscriptomics, proteomics, and metabolomics, to answer the classical questions, let's say, of who is present in a location at a certain depth at a certain time, which is the metabolic potential of organisms that are there and what they actually are doing with the expression data and the proteomics and metabolomics data. So far, so the project, as I said before, started in 2019, and it's still ongoing. As you can see here from the graph, not all parameters have been collected from the beginning, and there is a delay, let's say, of processing according to the nature of the data. So the sequencing of, I don't know, metagenomics is clearly more slow than processing the data from CTD, so just temperature and salinity, for example. So we have some dishomogeneity, let's say, in this kind of timing. What we have achieved in the last in this year, we published the first 10 months of data 1 year ago with a data paper published in Scientific Data, where we released the data for this first 10 months from April 2019 to January 2020. And we released data for the different sites and different depths and different dates, from physical parameters, nutrients, pigments, fluorimetry data, microscopy data, and also metabarcoding for prokaryotes and eukaryotes, and the metagenomics of prokaryotic plankton. For that, we relied on analysis that were done in collaboration with the Shinichi Sonagawa group in Zurich at the ETH, where they performed a single assembly of our samples and generated a prokaryotic gene catalog together with a set of almost 4,000 of metagenome assembled genomes. On the eukaryotic side, which is mainly my interest, we are currently undergoing analysis on 3 independent levels. So on one side, we have a set of, let's say, reference genomes and transcriptomes of species that have been collected and studied from the Gulf of Naples in culture. And for those species, we can simply map the metagenomic and metatranscriptomic reads against those reference genomes and transcriptomes to study the abundance, expression, and distribution of these target species in the Gulf. But on the other side, we are the other level of analysis is the eukaryotic MAG reconstruction and annotation in order to obtain a catalog of NEREA MAGs. And we also have the meta-T data for eukaryotes that we are trying to assemble in order to obtain a catalog of NEREA eukaryotic genes, similar to what has been done with Tara Oceans data with the MATU, or the Marine Atlas of Tara Oceans unigenes. So I'm going to talk a little bit more about this second step, because we are ready with that. So we started with the co-assembly of the data for the first 10 months. So it's 59 metagenomic samples. If you consider that we have different size fractions and that it's summed up to 59. And with those samples, we did a co-assembly to obtain the contigs. And then we performed the semi-automatic procedure to have first a set of meta bins through the automatic binning, so groups of potential mags that were a cluster of contigs that were put together according to their differential coverage and sequence identity. And with this set of meta bins, we then manually extracted and curated eukaryotic mags using Ambio as a software and the interface to do that. And we ended up with 52 eukaryotic mags that we profiled both taxonomically and functionally with gene prediction and gene annotation. And this work is ready. We are going to submit it next week, hopefully. And we found a variable amount of genes that were uncharacterized and assigned, but a decent amount of genes were assigned for each mag, considering that this is eukaryotic mags. One other thing that we did this year was to write a proposal together with collaborators from the Ohio State University, and specifically with the group of Matt Sullivan, which is interested in viruses, ocean viruses. And we won this proposal to integrate the ecogenomics, this new viral host linkage method, and the functional genomics to study a phage-host interaction using NEREA data. And we were happy of this proposal that we won. This will allow us to sequence 1 year of metagenomic samples for all size fractions of NEREA. We're now currently in the process of still extracting the DNA of these samples, so the results are not there ready, but we're anyway very, very happy about it. And also the last thing that I wanted to talk about was the link between field observations and lab experiments that we aim to achieve in NEREA. And last year, at the end of 2024, we started, there was a new PhD starting at the Stazione Zoologica with Erika Martone, this PhD student under my supervision, and other people from Stazione Zoologica. And the aim of the PhD project is to study the bloom dynamics of diatoms in the Gulf of Naples using all the omics that we collect at NEREA, but also integrating in lab experiments to target the biosynthesis, the genes involved in the biosynthesis of the domoic acid molecule, which is a neurotoxin that is produced by many species of the genus Pseudonychia. And Pseudonychia multistriata is a very well-studied species in the Gulf of Naples, for which we have genome and transcriptome and culture collections and so on. And this is a species, and this is a genus that is able to produce harmful algal blooms. So it has a huge impact for the ecosystem from an ecological, economical point of view, and so on. So what Erika is doing is, on one side, she's in the lab trying to cloning those genes and understand better the molecular mechanisms involved in the biosynthesis of this molecule, and which are the factors that influence and trigger the expression of those genes. And on the other side, she's looking at the metabarcoding data now for exploring diatom diversity in the Gulf of Naples with the NEREA data. She's seeing which are the SVs that are shared or not shared among the different sites, for example. She could see how Sarno and Canyon, for example, so the river mouth and the deep station are very different from each other in terms of shared ESVs. And this was also clear when she did a clustering approach where she clustered the samples according to SV's presence and absence. And she could see how the deep stations of the canyon here, so 719, 750 meters depth in the canyon, are clustered very apart from any other sample. She saw how the samples from Sarno, so when you see NRS, it is Sarno, and NRC, so the canyon, never clustered together. And overall, she could see that samples clustered together more according to sampling date than according to depth. So there is a strong signal on this. And she then looked at she's looking at the taxonomical composition at the diatom level at the site Marichiara, which is the one that is sampled each month. And she could see how sample communities varies with depth and season more than with size class. So here you have the different size class: microplankton, nanoplankton, picoplankton, and then on the x-axis you have the dates. There is a clear and consistent dominance of Chetoceros here in the light blue, and there is also in light orange here a presence consistent of what we define rare SV, so genera that have a lower relative abundance. So this is still preliminary, and she's going to analyze metagenomics and metatranscriptomics data as well to look specifically for genes involved in the biosynthesis of the domoic acid as well. So yes, so to conclude some consideration, we use NEREA, we see NEREA not only as a monitoring effort, but especially as a case study to test hypotheses, methods, and models that we can then apply also to larger scale, because we believe that this is essential as a multidisciplinary and integrative approach to address the complexity of marine ecosystems. But of course, this is full of difficulties and challenges. I just listed a few here, but there's a lot. So on one side, it's not trivial for us to go actually sampling each month on the boat in terms of people available, money, and logistics. Everything is not super straightforward for us, and we also have I think we are really not advanced in terms of metadata organization. It will be, in my opinion, for example, it would be amazing if we could have an infrastructure or something where we can actually directly put the data from our log sheet that we bring on board to something that can be accessible for the whole community of NEREA and something like this. We are still exchanging Excel files between labs and with all the difficulties that this implies. And of course, we have issues in the high cost of sequencing efforts, especially with the meta-T and especially with both prokaryotic and eukaryotic meta-T actually. So for example, we won this proposal with JGI, which will cover 1 year of data, but we then have other fundings for other years. With sometimes we have holes, some months are not covered yet, so we're always in the we don't want to interrupt the sampling, because this is also the strength of the project, is that we do it continuously. But on the other side, we are accumulating samples that we are not sure when and how we can actually sequence them. And then once we have them sequenced, we also have an issue in computational demands of bioinformatics and downstream data analysis, storage of data, and actually who's gonna actually look at the data and do some science out of it. So it's a flood of data that we also sometimes need to look at with calm. So I mean, that's just a few challenges that we encountered during these years. And yes, I think this is it. I want to thank all the people involved in the project, especially the coordinators of NEREA, and also Raffaella, who asked me to give the talk today, if I was available to give the talk today, and Antonio Longo and Erika Martone, the PhD students that are actively working and actually making the science out of NEREA. And helping with the sampling and everything, and also there's many other people involved, and thank you all for your attention.
Thank you very much. It's great to see the results from the NEREA Observatory now, and it looks like there's some common challenges among the different OBON projects, and also some offers to help.
Yeah, and I forgot to mention that also Mare Chiara is a station that is part of OBON as well. So in parallel, we go on board, and we do each 2 months we do both samples, both filtrations. So there are some differences, but still ongoing there. Yeah.
Okay, that's great. And I also shared the publication that you mentioned. Hopefully it's the right one, from your 2024 publication.
Yeah.
In the chat.
Thanks.
Is there a quick question before we wrap up? There's a question in the chat regarding the preservation of the filter. What preservatives do you use for multiple studies?
Sorry, I don't see.
Specifically for the analysis of DNA and proteomics and metabolites, proteome metabolite together.
Being more specified, you process sample for the analysis of DNA and protein both together. No, it's different filters, if this is the question. So we collect filters for DNA and RNA. Well, at the beginning, we had one single filter because it was Genoscope that was sequencing our data. And Genoscope could extract simultaneously DNA and RNA from the same filter. Because they had this machine, this cryogrinder that was able to do that. We don't have this machine, so now we are dividing the filter in two so that we can extract DNA and RNA separately. And then we collect other filters for protein and metabolites, if that was the question. I don't know if I answered.
I think so. Thank you.
Okay.
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 Thank you very much. And I'm sure these conversations will continue next week. I know Raffaella isn't able to attend in person, but if she's able to attend, or if you'd like to attend some of it online, Lucia, that would be very welcome.
All right.
So thank you very much again to all the speakers. Some really fascinating presentations. And look forward to seeing some of you in Hobart next week.